Hi, everyone. First of all, I want to thank Massimo and Guilherme for the last-minute invitation for this talk. I'm very happy to be here and present to you some results of our conservation genomics of the Brazil nut tree, or the Amazon nut tree, the most correct name, I think. The Brazil nut tree is a deciduaceae that's widely distributed in the upland forests in the Amazon. It produces the delicious and the nutritious Brazil nuts, and these nuts are almost exclusively obtained from native forests by local communities. So the commercialization of these nuts represents an important source of income for thousands of people in Brazil, Bolivia, and Peru. Today I'm presenting the results of two ongoing projects that I'm leading. The Brazil Nut Genomic Project aimed to produce genomic resources for the underrepresented tropical trees. And then we assembled and annotated the Brazil nut tree genome. And then we use this genome to do this NIP calling for this other ongoing project that it's aimed to study the conservation genomics of the Brazil nut tree across the Brazilian distribution area and also the Peruvian area. To assemble our genome, we used PacBio long reads with a coverage of 108x and did the scaffold with Hi-C. The estimated size of our assembly is 605 megabases, that is a small genome, and is quite similar to this flow cytometry estimated of 607 megabases. And more than 99% of our genome is in 17 chromosomes, and it shows that, are very proud to say that we achieved reference quality genome. And as an example, the Contig N50 of our assembly was 10 times greater than the mean Contig N50 obtained from 800 genomes. 40% of our assembly are repetitive elements, and we have very few events of recent duplications in the genome. You are just finishing writing this manuscript and expect to submit very soon. These are the colleagues who are working in this Brazil Nut Genome Project, and they are from different institutions in Brazil. The financial grant was provided by FAPESP, that's São Paulo Research Foundation. So after telling very briefly about our genome, I enter in the title of this talk, and I'm going to presenting two studies. The species distribution modeling study, where we generated maps, suitability maps for the Brazil nut tree at present and in two future scenarios of climate change, and some results of the genetic diversity and structure that we sampled 271 trees in 47 locations in Brazil and Peru, and then obtained RADSEC libraries and produced the sequences, single-winged reads. This NIP calling was done with BB tools and our freshly new reference genome, and after filtering, we had 8,000 diallelic SNPs. It's not a trivial task to study genetic diversity of a widely distributed species like the Brazil nut tree, especially if the species is distributed across a forest, a continual forest. It's not easy to transit in the Amazon by boats or by roads. It's a challenge. And also several factors may influence the Brazil nut genetic structure. This species is not an easy one to study. It occurs in a large environmental variation, as shown in this climate typology map on the left. And also, the historical demography is very complicated. In that central map is a habitat suitability map produced by Thomas and collaborators, and it shows the suitability of Brazil nut during the last glacial maximum, and it shows that the contraction of populations of Brazil nut tree was very strong. And then in the right, it shows the distribution, two things, the habitat suitability of Brazil nut tree, the green colors, and then the areas, the regions with a high concentration of geoglyphs here in the western region, and the anthropogenic dark earth. So these are regions where we believe that human populations in the past, pre-Columbian inhabitants, occurred in high concentrations with large numbers of populations with several peoples. And then there is a more recent theory that these populations have influenced, promoted, and dispersed several species. And Brazil nut tree is one of them because because of your nuts. For the SDM study, use almost 1,000 presence points and 35 explanatory variables. And the novelty of this study is including these records of geoglyphs and the anthropogenic dark earth as explanatory variables for our models. And this bar plot shows that these three climatic variables that were most important for our best model. So precipitation, temperature, seasonality, and precipitation of the coldest quarter. But the plot also shows that the dark earth sites and the geoglyph sites were also important in influencing the habitat suitability of Brazil nut tree in the present. This is our best model. I'm not going to details of the statistics we used to generate this, but I want here to thank you, Gabriel, Everett, Viviana, Tobias, and Diana that helped producing these maps. And these are the two future scenarios of climate change. On the left, the optimistic one, and in the right, the worst case scenario, and they are quite similar. And in both, you can see that there are several areas that will become unsuitable in the future, the ones that are in red. And this map shows the distribution of our sample locations. We sampled 271 trees, 47 locations. The number of trees per site is 2 to 12 in an average of 6. Our first analysis was identifying FST outliers using SNMF and tests, and then we found 110 candidates. In the left is the distribution of allele richness in these 118 SNPs, and then it's evident that for these candidate SNPs, the central area shows higher genetic diversity. And in the right, it shows a higher concentration in the easts of locally common alleles that are alleles that occurs in high frequency, but only in part of the sites. So SNPs that are geographically strict to some regions, in this case, in the East region. After excluding these SNPs, you used the neutral SNPs to study genetic structure, again using SNMF and TESS, and both produced the same results that are 5 ancestral populations, and here are the distribution of these genetic groups across the landscape. There is a strong genetic structure, and those populations or the plants that are more in the central area of the distribution are more admixed. And this plot, this slide combines the results of genetic structure and genetic diversity using these neutral SNPs. The colors represent the the 5 geographical groups, genetic groups, and the numbers are average estimates of observed heterozygosity for each one of these groups. So the populations here in Southwestern, that represents the yellow and red groups, had on average higher genetic diversity than those in the East area. And this final map combines the SDN map with the genetic groups. So just exemplifying the worst case scenario, we see that the light blue are the areas who are going to become unsuitable, and you see that the purple genetic group, the north part of the red group, the pink, blue, and the blue are those that tend to be lost. And finally, I want to thank all the colleagues that are working in this project. There are people from different institutions in Brazil and South America. So thank you very much.